intro about what transcriptomics are. So it's basically, you can go to the next slide, Angie. It's just the study of RNA transcripts in a biological sample. This is really useful for a lot of different applications. Most of the time it's to study and understand the gene expression in biological systems. And a lot of what we do is also looking at how cells or organisms respond to their environment. And a good way to do that is to use transcriptomics. And it can also assist with biomarker discovery and gene function annotation. Next slide. So what does a typical transcriptomics workflow look like? So the first step is always to grow or culture or collect your samples that you're interested in. Then the next step is to extract the RNA from those samples. And then the next step is to do some quality control, make sure your RNA looks good, and then prepare the RNA for sequencing libraries. Once those are prepared, you can run them on different types of DNA sequencers. So most of what we do is on the Illumina platform. And from there, then you go on and analyze data using some high performance computing systems. Next slide. So I just want to go over quickly what types of equipment and capabilities we have over here. So we're all equipped to do all of the sample prep and quality control steps. I'm just highlighting this MP Biomedical's cell homogenizer. So this is really useful for breaking apart difficult samples. We have things like nanodrops and qubits to do the nucleic acid quantification. We have a tape station to do some quality control on the quality of these nucleic acids and to visualize them. We're starting to increase our throughput. So we have some new liquid handling tools to do automation that allows us to do more samples and process more samples simultaneously. And then additionally, we haven't really used this, as far as I know, in any ABF projects, but we've used it for other transcriptomics experiments. This is single cell sequencing using this 10x genomics platform. And this allows a really high resolution understanding of how cells are behaving at an individual single cell level. Next slide. We also have a lot of options for sequencing on site here. So we typically do most of our transcriptomics doing RNA-seq using these short read sequencers on the Illumina platform. For small scale experiments, we use a MiSeq. For mid to large scale experiments, we usually use, we just got this brand new NextSeq 2000 instrument. We also have some long read sequencers like the PacBio instrument and some Oxford Nanopore options. And these can be good if you're interested in understanding things like RNA structure or alternative splicing or expression of different alleles. We also have a lot of options to actually do the sort of data storage and data processing and analysis that you need for these large data sets. So we have a lot of high performance compute clusters that are available as well. Next slide. And just a few example use cases for what transcriptomics can be used for. These experiments were all done with a yeast, Rhodosporidium, that's used for biomanufacturing applications. One thing that you can do is look at gene expression for genes that you're interested in under different growth conditions. You can look at how these transcriptomes change over time. And then you can also look at, if you have different samples or different conditions, you can identify the genes that have differential expression levels between those conditions or samples. Those are kind of the most common use cases we typically do.